No water, no problem. Drought Survival in the African Lungfish by Caden Purnell from Oregon State University's College of Science. The African lungfish of the genus Protopterus inhabits freshwater environments where oxygen conditions are low, daytime temperatures are high, and seasonal drying is prolonged. To cope with life-threatening hypoxia and drought, African lungfish obligately breathe air through a set of lungs and, as water dries up, induce estivation. For fish, living out of water presents significant challenges that often prove lethal. To overcome this, the African lungfish has developed adaptations combating dehydration, energy depletion, and toxicity that allow it to estivate without food or water for years on end. I will be discussing the African lungfish's unique ability to maintain osmoregulation, metabolic control, cytoprotection, waste management, and cardiorespiratory control during estivation. For most fish, living out of water simply isn't an option. However, the African lungfish can prevent dehydration by secreting mucus that hardens into a leathery cocoon and burrowing in the mud. In addition, these fish have a reduced gill size and slender body surface, which facilitate a relatively low permeability to water and ions. Changes in the expression of pituitary hormones linked to the maintenance of estivation, like prolactin and growth hormone, occur to control osmoregulation as well. Control over metabolic activity to conserve limited energy stores during estivation involves complex interactions between up and down regulation of the expression of many genes to reduce brain activity, suppress the proliferation of brain cells, reduce glycolytic capacity, and subdue the energetically expensive synthesis and degradation of protein. To protect cells from cold and oxidative damage, proteins associated with cold tolerance and intracellular antioxidant enzymes are synthesized at increased rates, boosting cold and oxidative stress resistance. RNA binding proteins, glucose-regulated proteins, and superoxide dismutase are some of the key biological molecules involved in this process. During estivation, ammonia concentrations increase. Thus, to protect neural tissues from ammonia toxicity and curtail contamination of its internal environment, the African lungfish must maintain a slow rate of waste generation and detoxify ammonia to urea. Consequently, urea concentrations increase significantly in the muscles, liver, and plasma of the African lungfish, causing water content in muscles to simultaneously increase, and thereby demonstrating that muscles act as a water reservoir during estivation. In fact, after 5 months of estivation, muscle urea can increase up to 13-fold, and plasma urea can increase up to 8-fold without causing any disruption to blood osmotic equilibrium. Estivation in the African lungfish involves a reduction in heart rate, blood flow, blood pressure, and oxygen consumption, whereby obligatory air breathing is the exclusive source of oxygen. This reduction in cardiac performance is achieved through changes in the distribution and concentration of nitric oxide, an endogenous gas that causes vasodilation, and a reduction in mitochondrial respiration in African lungfish. During estivation, an enzyme responsible for producing nitric oxide, termed nitric oxide synthase, are upregulated in the kidneys, heart, and lungs. Simultaneously, the same enzyme is downregulated in the gills. Through these changes, the African lungfish maintains cardiac efficiency while slowing oxygen consumption and cardiorespiratory work. The ability to detect environmental cues and respond accordingly is a vital trait for survival in all animals. The African lungfish's proficiency in appropriately responding to changing environmental conditions, like drought, has clearly proven integral to its persistence. The arduous challenge of surviving out of water has been shown to be impossible in most fish species. Yet, the African lungfish's unique ability to make behavioral, physiological, and biochemical changes to survive extreme environmental challenges has allowed these fish to persist for millions of years. Therefore, African lungfish are extremely well adapted to surviving out of water and without food, up to 7 years. Through its strong control over osmoregulation, metabolism, cytoprotection, waste buildup, and the cardiorespiratory system, the African lungfish has demonstrated itself as an animal that reigns in evolutionary fitness. Although extensive research has been done on the African lungfish, there is still much that we do not understand about these animals. For example, the African lungfish is known to be a highly important group that retains many ancestral genes and is closely linked to tetrapods, yet many of its genes are still poorly understood. Thus, its genome should be mapped so that scientists may better understand the role that proteins play in estivation. In addition, the mechanisms by which African lungfish osmoregulate during prolonged estivation is still not fully understood and needs further investigation. Thank you.